we're disrupting the process for cancer cells to grow, whether it's KRAS driven or whether there are other mutations. This just broadens the opportunity for our ADC payload in ways that other ADCs do not have, all because of our payload and its unique biology of targeting cancer.
Welcome back, and thank you for joining us for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator. Today we are featuring Akari Therapeutics, and I am very pleased to be joined by Abizer Gaslightwala. He is Director, President, and CEO of Akari Therapeutics. Welcome, Abizer.
Thanks, Janine. Thanks for having me today.
Always happy to have you. And before we get started, I want to remind our audience that Akari is publicly listed on NASDAQ and trades under the ticker AKTX. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at akaritx.com for the latest information. Abizer, as I mentioned, we are so happy to have you today. And so for our audience, today's segment is going to focus on the recent data that was accepted at ASCO, which is a preeminent scientific congress in the oncology space. So we're going to dive right in. And Abizer, I have to first congratulate you and your team. This is the first ASCO abstract accepted for Akari. Very impressive.
Yeah, thank you. We're really excited about it as well.
Awesome. Well, I guess let's start with summarizing the data. That would be helpful.
Yeah, sure. So what was really, I think, significant and monumental and why we got this abstract accepted and released was because of the role of the cancer tumor biology and the unmet need. And so this experiment was actually done in a very difficult set of tumors that are driven by a mutation called KRAS. KRAS is one of the most well-known difficult targets to go after in cancer. It's been a target known for 30 years. There have been a couple of KRAS inhibitors approved, if you follow the space, by Amgen and Bristol Myers Squibb a couple of years ago, but those have been, fairly moderate in their uptake and their overall results. And so I think one of the things that's happened in the field is to say, what's next? What's better? What can be done to improve the outcomes for these difficult cancers that are driven by KRAS? And some of the most difficult cancers that are driven by KRAS are lung, colon, and pancreatic, all three very big unmet needs still when you look at the cancer space. So as that backdrop, what this data showed is that, as we've been saying with our novel RNA splice targeting payload for ADCs, we believe there's a unique mechanism that applies to many different tumor types and many different pathways. And in this data set, we actually showed that it actually applies to KRAS, not just as a single agent, but importantly, synergistically in combination with some of those current KRAS inhibitors that I mentioned, and in particular, adagrasib, which is the Bristol Myers Squibb compound. That was a compound that's currently on the market. And we actually showed data that shows we have synergy when we add our ADC to adagrasib in these difficult cancer cell lines, notably pancreatic. Why that's important is I kind of talk about synergy. Synergy is not just 1 + 1 = 2. That's additive. Synergy is 1 + 1 = 4. And so as we did the experiment, we showed that when we add our ADC with our novel payload to adagrasib, the commercially approved product, we get that 1 + 1 = 4 or 5, or that synergy. Importantly, what we showed is that other ADCs that use conventional payloads, they don't even get 1 + 1 = 2. They get 1 + 1 = 1.5, or even lower than that. That's called antagonism. So they're not just adding, they're actually taking away. So for whatever reason, those ADCs and their conventional payloads are detrimental to the KRAS inhibitor that was tested at adagrasib. So you're seeing kind of opposites here. You're seeing our payload be synergistic with current standard of care, whereas the other ADC payloads are being antagonistic. That was what was really unique and monumental around this experiment.
Well, not only monumental, but absolutely encouraging. So let's break this down a little bit further, because I think this is so important. And what you said is, I feel like incredible progress, not only in the space, but for Akari. So what stands out and what are the key takeaways from this data in the simplest form?
Yeah, what I would say is that this continues to validate our payload, the PH1 payload that targets RNA splicing, has a broad therapeutic benefit potential for many different cancer tumor types, whether they're solid tumor or liquid tumor, or whether they're driven by really difficult mutations like KRAS. That's not true for many other ADC payloads because their biology does not necessarily make them capable of working in these very difficult tumors driven by mutations like KRAS or others. That's unique to our payload. And so, what this is showing is that even in the most difficult tumor types, we have a payload that works because of the novel biology. And so, think about it this way: because we hit a central processing vehicle for all cancer cells, RNA processing, we're disrupting the process for cancer cells to grow, whether it's KRAS driven or whether there are other mutations. This just broadens the opportunity for our ADC payload in ways that other ADCs do not have, all because of our payload and its unique biology of targeting cancer.
Excellent. Extremely helpful, Abizer. As I mentioned, very exciting for Akari as well as in this space. So, what do these data mean for AKTX 101's development program?
Yeah, it's really exciting because it adds to the what I call concentric rings of opportunity for AKTX 101. We're still focused on bringing our TROP2 ADC with our novel payload into going after urothelial bladder cancer. We're focused on trying to get into the clinic as soon as we can, and I'll speak to that in a second. But this just opens up additional opportunities. So now, as we think about establishing that beachhead in urothelial bladder cancer, where else should we go? We have opportunities in breast cancer, obviously. Now we think about lung cancer, maybe KRAS lung cancer, maybe things like pancreatic cancer with KRAS mutations. It just continues to add and increase the concentric rings of opportunity that we have with this asset in ways that other ADCs that target TROP2 don't. That's all driven by the payload's effects.
Wow, so it's an opportunity really to kind of build out your pipeline here with a lot of indications and opportunity.
Yes, I mean, it's pipeline and a product in a sense. And we've always said this payload can create many ADCs. For this ADC, the TROP2, we believe we can create multiple pipeline opportunities from the same ADC as well.
Excellent. Well, what a way to end this segment, Abizer. Congratulations on your progress. Can't wait to have you back. And with that, this does conclude our What This Means segment with Akari Therapeutics. Again, Abizer, thank you for joining us today. And to our audience, thank you for your time and attention. And if you like what you saw today, I encourage you to visit akaritx.com for more information on the company and to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. And I encourage you to do so because this announcement was certainly exciting, and it seems like there's more to come this year. And as a reminder, Akari trades under the ticker AKTX. Abizer, thanks again, and wishing everyone a great rest of your day.